a topic by name gametogenesis. Now, what is gametogenesis? Genesis is the making or the synthesis, the production of gamete. So, gametogenesis involves two words. One is the gamete, the other is the genesis. Genesis means the synthesis of, gamete is the haploid cell. The synthesis of or the making of the production of the gametes can be called by the word gametogenesis. In the world of zoology, there are several animals which have sexual method of reproduction to produce the progeny. Now, if that is the case, then what is the methodology involved in making the next generation? All the sexually reproducing organisms have the gonads. It could be a male gonad or the female gonad. Now, within the gonad, the fundamental layer which is essential for the making of the gametes is the germinal epithelium. That means within a gonad there will be germinal epithelium. So, the germinal epithelium present within the gonad will do or perform several steps and ultimately resulting in the production of a haploid cell by name the gamete. So, I repeat the sequence again: gonad containing the germinal epithelium after several steps, which includes the meiosis also, will result in the making of the haploid cell called the gamete. Now, if the gonad is the male gonad called the testis, the germinal epithelium after gametogenesis called spermatogenesis will produce the male gamete called the sperm. If it is a female gonad, the ovary, the germinal epithelium aims at producing the female gamete, which is also called the ovum or the oocyte. That means the male gonad after the spermatogenesis will result in the production of the sperm, and the female gonad after the process called the oogenesis will result in the making of the ovum or the oocyte. That means two haploid cells are produced in the male and the female reproductive organs called the primary sexual structures by name the gonads. Once the gonad makes the gamete, the haploid structure, these two cells, the male and the female gamete, will unite. This union is called as the fertilization, also called the syngamy, and that results in the making of a diploid cell called the zygote, and from where the life will unfold. Now, today let us now understand the fundamental steps involved in the process called the gametogenesis. Whether it's a spermatogenesis or the oogenesis, the three important steps are, look at the board and we can understand the idea. The gametogenesis involves three important phases. The very first one is called the proliferation phase, second is called the growth phase, and the third one is called the reduction phase, which involves the reduction division by name the meiosis. అంటే గెమెటోజెనిసిస్ అనేటువంటి ప్రాసెస్లో మనకి ముఖ్యంగా మూడు స్టెప్స్ ఉంటాయి, అండ్ దే ఆర్ ద ప్రాలిఫరేషన్, ద గ్రోత్ అండ్ ది రిడక్షన్ ఫేజ్. అయితే ప్రతి ఒక్క ఫేజ్కి కూడా ఒక ఇంపార్టెంట్ ఫీచర్ ఉంటుంది. ప్రాలిఫరేషన్ ఫేజ్లో వీ విల్ సర్టన్లీ సీ దాట్ ద సెల్స్, ద మదర్ సెల్స్, ద జర్మ్ మదర్ సెల్స్, దే విల్ పర్ఫార్మ్ ది మైటోటిక్ డివిజన్స్. దానివల్ల ఆ సెల్ నంబర్ ఇంక్రీజ్ అవుతుంది. సో in the very first part called the proliferation phase, the mother cell, the germ mother cell performs the mitotic divisions several times, and that results in the production of numerous germ mother cells, and some of them do increase in size, and that increase in size of that mother cell can be called as the growth phase. Once the mother cell completes the growth phase, the diploid mother cell after the completion of the growth phase will enter into the next phase, which is called the reduction phase. The reduction phase will involve the meiosis 1 as well as the meiosis 2. Sometimes we call them as the maturation divisions also. So the meiosis 1, meiosis 2, after its completion ultimately results in the production of the gametes, And these sequential activities or events which occur within the gonad, whether male or female, is called as the gametogenesis. So, gametogenesis primarily involves 3 steps, 3 phases, and they are the proliferation, the growth, and finally the reduction phase. And after reduction phase is completed, there will be production of the gametes which are haploid. Now, let us study the 2 different types of gametogenesis, one in the male, the other one in the female. In the male, it is called as the spermatogenesis, and in the female, it is called as the oogenesis. So, let us start our learning with spermatogenesis, which takes place in the male. So, we are learning the steps involved in spermatogenesis. The very first question is, like, where is this particular process or biological activity going on in a human male? We all know that the male gonad is called as the testis, and within the testis we will have this lobules, and in the lobules we find the seminiferous tubules. So, within the seminiferous tubules, the lining is called as germinal epithelium. So, as I was previously referring to, gonad should have the germinal epithelium, and the germinal epithelial cells will pass through 3 different stages and ultimately result in the male gametes or the sperms. Now, if you look at the sequence here, male gonad, also called the testis, containing lobules, and the lobules will have the seminiferous tubules, and the seminiferous tubules are lined by the germinal epithelium. Now, interspersed between the germinal epithelial cells, suppose this is the seminiferous tubule, we know that in between we will find these specialized cells called the Sertoli also. So, the Sertoli cells do have a role at the ending of the spermatogenesis. So, the exact location or the site where the spermatogenesis takes place will be testis, which contains the lobules containing the seminiferous tubule, and within the seminiferous tubule we find a special cells, a special epithelium called the germinal epithelium, and in between the germinal epithelial cells we find the nurse cells or the Sertoli cells. But the spermatogenesis is done by the germinal epithelial cells. The germinal epithelial cells, we find a layer of epithelial cells called the germinal epithelial cells, which are involved in the process called spermatogenesis. Of course, the role of hormones is certainly to be understood here, which we will be dealing after some time. So, I was talking about the germinal epithelium. So, the germinal epithelial cells are all basically diploid. So, the male gonad will be having a layer of cells which we call it as the germinal epithelial cells. Now, our focus will be on the germinal epithelial cells and how, under the influence of hormones, these germinal epithelial cells begin the process called spermatogenesis. About the role of hormones we will discuss later. Let us start the sequential biological activity called the spermatogenesis. Now, I have written here germinal epithelium, where all the cells of the germinal epithelium will be basically the diploid cells, each one consisting of about 46 chromosomes, and we generally write it as 44 plus X, Y. Now, what is the name of the cell which is commencing the process called spermatogenesis? The name of the cell will be called as spermatogonium. So, we call it as spermatogonium. So, spermatogonium is the cell. The singular is spermatogonium, the plural is spermatogonia. So, we have taken a particular cell, the mother cell, the sperm mother cell, the spermatogonium, consisting of about the 46 chromosomes. Now, this group of cells, the spermatogonial cells, the diploid cells, the mother cells, now these cells primarily enter into a process called mitosis. So, they do continuously the mitosis, increasing the number. We call this step as the proliferation. As we have discussed earlier, the three important phases of the gametogenesis, one being the proliferation phase. Now, when I say the spermatogonium is repeatedly performing the mitosis, it means it is in the proliferation phase, so that the number of cells will be increased. And occasionally, now when some of the primary this spermatogonial cells will be becoming the primary spermatocyte. So, the very first step is the multiplication of the mother cells. The mother cells are diploid, they perform the cell division called the mitosis, and because the number is increasing, we call it as the proliferation phase. Now, some of the spermatogonial cells will enter into the growth phase. So, when a spermatogonium singular will enter into the phase called the growth phase, so naturally the dimension of the cell will increase, and this will be remaining as diploid, but the size of the cell will increase, and we call it as the primary spermatocyte. So, the primary spermatocyte is the result of the growth phase. Now, this primary spermatocyte will be entering into the meiosis 1 and also the meiosis 2, and finally producing the spermatids. Spermatids undergo change to become sperms. We call it as the spermiogenesis. So, let us see the next step. I am writing it here, the primary spermatocyte. The primary spermatocyte is completing the meiosis 1. So, if it does that, the post maturation division, the meiosis 1, we get the two cells which are haploid. So, this is N and this is also N, and we call them as the secondary spermatocytes. So, now we have haploid cells here called by the name secondary spermatocytes. So, primary spermatocyte, which I have rewritten here, after meiosis 1 will produce two cells which are called as the secondary spermatocytes. The secondary spermatocytes are haploid. And then what about the meiosis 2? Who will enter into the meiosis 2? The two haploid secondary spermatocytes will enter into the meiosis 2 and they will produce spermatids. So, let us see that step. So, each one will enter into the meiosis 2 and will produce the cells which are called as the spermatids, and naturally these are also the haploid and they also will give two spermatids. So, let us understand the steps here, the sequence. The first one is the spermatogonium becoming primary spermatocyte. The primary spermatocyte after the meiosis 1 will give the two haploid secondary spermatocytes. The two secondary spermatocytes will enter into the meiosis 2 and will produce 4 spermatids which are also haploid. Now, if you look at the design and structure of the cell, they are all spherical, they are round, and they are all the haploid cells. Now, a very important step wherein the spermatid will undergo a morphological, physiological differentiation resulting in a very well designed gamete called the male gamete. What is this morphological change, a physiological change which is done by the spermatid? This spermatid undergoes a very important phenomenon called spermiogenesis. So, the transformation or the structural, morphological and physiological change of a spherical spermatid into a functional designed gamete. This step is called as spermiogenesis. That means all the 4 haploid spermatids will undergo the spermiogenesis and produce a well-designed structure called the sperm, the male gamete, the male sex cell, or the spermatozoon. That is what I am writing. I am selecting one spermatid to indicate that particular change. The spermatid will undergo that process called as spermiogenesis and ultimately making the male gamete called as the sperm. Now, once the sperm is made, we can say that the total spermatogenesis is completed, and once the spermatogenesis is completed, the final activity which goes on within the seminiferous tubule will be the Sertoli cell, which I have already spoken. The seminiferous tubules will be internally lined by the germinal epithelial cells, and here and there we find the Sertoli cells. The Sertoli cells or the nurse cells will be releasing away the well-formed sperm. That means after spermiogenesis, from the Sertoli cell, the sperm must dissociate itself, move away and part from the Sertoli cell. What is this parting away, moving away, the separation, the segregation from the Sertoli cell called? This is called by the name spermiation. That means after spermiogenesis, the next event is called as spermiation. Once the spermiation takes place, the journey of the sperm begins, and the sperm will move its way or make its way from the seminiferous tubule and gets into the next structure, which is called as the rete testis. And from the rete testis, it goes into the vasa efferentia and gets stored in the epididymis. So these are all the various events pertaining to the spermatogenesis, and after which the sperms are stored in a structure called as epididymis. Now, let us study the hormonal effect on the phenomenon called as the process called as the spermatogenesis. Now, let us learn the effect of hormones on the process called spermatogenesis. As I have depicted here, presented here the sequence of activity, look at this. Now, hypothalamus, we know that the floor of the diencephalon is called by the name hypothalamus. A very diversified structure which will control many functions in the body, and this area called the hypothalamus will have a specialized cells called the neurosecretory cells. The neurosecretory cells in fact release the neurohormones, and the neurohormone which I am referring here is called the gonadotropin releasing hormones. So, when you say gonadotropin releasing hormone, these are small sized hormones, and these chemical messengers which are neurohormones released from the neurons of the hypothalamus will be transported by a very specialized portal system which is called the hypophyseal portal system. The hypophyseal portal system is located in the pituitary stalk or the infundibulum. Now, let us understand the sequential activity regarding the hormonal effect. The hypothalamus, the neurosecretory cells release the neurohormone by name the gonadotropin releasing hormone. This gonadotropin releasing hormone will enter into the blood of the hypophyseal portal system which is located in the pituitary stalk or the infundibulum. After completing its very short distance journey, the gonadotropin releasing hormone will be acting on the adenohypophysis. When the gonadotropin releasing hormone acts on the adenohypophysis, which is the target for gonadotropin releasing hormone. So, this gonadotropin releasing hormone will act on the adenohypophysis, and this component of the pituitary gland will release the gonadotropins by name the follicle stimulating hormone and the luteinizing hormone. That means the hypothalamus is influencing the production of the gonadotropins. The gonadotropins are follicle stimulating hormone and the luteinizing hormone. Now, let us see the working of these two chemical messengers separately. The adenohypophysis is releasing the luteinizing hormone. The luteinizing hormone is also called by another name, interstitial cell stimulating hormone. Now, with the luteinizing hormone is otherwise termed as interstitial cell stimulating hormone, this LH or 𝗖𝐶𝗺 will be entering into the blood plasma and is transported to the interstitial cells. The interstitial cell or the Leydig cell or the interstitial endocrinocytes present in between the seminiferous tubules of the male gonad will be influenced by this particular hormone called 𝗖𝐶𝗺. That means the LH or 𝗖𝐶𝗺 will be performing a long distance signaling and endocrine signaling and will act upon the cells called the interstitial cells. The interstitial cells, the Leydig cells, the interstitial endocrinocytes are affected by the LH, and as a result the male sex hormone called the androgen, also called the testosterone, will be produced, and the androgen or testosterone will help in the process called as spermatogenesis. And now let us see the effect of the follicle stimulating hormone, which is another gonadotropin. Another gonadotropin by name the follicle stimulating hormone will be acting upon the Sertoli cells. The Sertoli cells or the nerve cells are influenced by the follicle stimulating hormone. The gonadotropin by name FSH, when it acts on the Sertoli cells or the nerve cells within the seminiferous tubules, it will promote the releasing of the making of some factors which will influence the process called spermiogenesis. So, let us recall the sequence: hypothalamus, gonadotropin releasing hormone, hypophyseal portal system, adenohypophysis, the gonadotropin by name FSH and LH will be released. The LH is also called interstitial cell stimulating hormone, which is performing the long distance signaling, the endocrine signaling, and acting upon the interstitial cells and the release of promoting the release of androgen or the male sex hormone, that is testosterone, which promotes the spermatogenesis. FSH, another gonadotropin, will act upon the Sertoli cells and will be influencing the process called as spermiogenesis. So, this is about the effect of hormones on the process called the spermatogenesis. Students, we are trying to understand a representation here, a picture which depicts the anatomy of the testis and also the process called spermatogenesis. Now, here within the testis we find the coiled seminiferous tubules. Seminiferous tubules are lined by the germinal epithelial cells and here and there the Sertoli cells. Now, further magnification of the structure, we can see the Sertoli cell, and here are the cells which are in the various stages of development, ultimately becoming the sperms. Here is the germinal cell, the diploid cell, which is of course entering into the proliferative stage, the growth phase, and finally becoming the primary spermatocyte, which after the meiosis 1 is giving rise to the two secondary spermatocytes, and both are haploid. The two secondary spermatocytes which are haploid after meiosis 2 will become the spermatids. So, I repeat, secondary spermatocytes after meiosis 2 will produce the spermatids. Each one will produce 2, therefore, the total number of spermatids are 4. The spermatids will undergo a morphological change, and that process is called as spermiogenesis, and finally becoming the sperms which are released from the Sertoli cell. That releasing is called as spermiation. The second component of our learning today will be about the process called oogenesis. As the name is suggesting, oo is standing for egg, genesis is the production. So, the process of producing the egg, ovum, ooted or the female gamete can be called as the oogenesis. Before we understand the different follicles, the primordial to the Graafian follicle, we will be learning the sequential events related to this oogenesis. Now, to understand the process of oogenesis, we need to first understand that there is a diploid cell which is called as the oogonium. Now, we all know that when we draw a comparison between the two events, the spermatogenesis and the oogenesis, the terminology between the two processes will be almost a kind of similarity will be observed. Like when spermatogenesis is being learnt, we use a word called spermatogonium. Here we will use the word called the oogonium. So, the oogonium is like a egg mother cell. It is a diploid cell and containing 44 plus 2 X chromosomes, and that makes it 46 chromosomes. Now, this oogonium, which is a diploid cell, will be increasing in size. When the oogonium is increasing in size, we know that it is the growth phase. We have already termed it as the growth phase before, the second part of the gametogenesis process. So, oogonium will increase in size and we call this as the primary oocyte. So, the primary oocyte is again a diploid structure and this one will be entering into the meiosis 1. So, when the primary oocyte is entering into the reduction division, the maturation division which is the meiosis 1, the primary oocyte will produce the secondary oocyte and a polar body. Now, this is what is called as a polar body and this one is called as the secondary oocyte. Now, both are haploid. Both are haploid. What is to be noted is the polar body is haploid, secondary oocyte is also haploid. That means the primary oocyte after the meiosis 1 will produce 2 haploid cells called as secondary oocyte and the polar body. Out of these two, there is unequal cytokinesis and for that reason you can see an unequal sized haploid cells. And moreover, the secondary oocyte continues its further life, further processes, that is maturation division 2, whereas the polar body may continue with the meiosis 2 or sometimes may disintegrate. So, it all depends. So, the point here is the primary oocyte after meiosis 1 will produce the secondary oocyte and the polar body. Both are haploid, but because of the unequal cytokinesis, we can see the distinction or difference in the size of the cell. The one which is essential, the one which is needed is the secondary oocyte and the polar body, which is called the first polar body. If it continues the meiosis 2, produces 2 more polar bodies which will certainly disintegrate. So, this is one is required, that is secondary oocyte, and suppose it does the meiosis 2, we get the ovum and other one called the second polar body. That means secondary oocyte after the meiosis 2 or the second meiotic division, maturation division will produce again 2 haploid cells, of which this one is called the ovum or the ootid and the female gamete, the female sex cell, and this one is the second polar body, and this is also unequal cytokinesis and this second polar body will disintegrate. The secondary oocyte certainly will do the meiosis 2, especially after the entry of the sperm, it will do the meiosis 2 and will be producing the ovum and the second polar body. Now, what are the cells which are definitely going to disintegrate in this process? This polar body, even if it does meiosis 2, the products, the 2 other polar bodies will also disintegrate and this second polar body will also disintegrate. That means from one oogonium there will be production of one ovum, one oogonium. After the completion of oogenesis will produce only one ovum or one oocyte. Now, I am relating this oogenesis with the follicular development. Now, in a human development or human development of the female gamete, the ovum, what is a very noticeable feature is the primary oocyte. The primary oocyte will be in such a stage wherein it can complete the reduction division 1 quite comfortably, quite easily, because the primary oocyte will be in the stage where the karyokinesis, where it will complete the prophase 1. So, that means the primary oocyte completes the prophase 1 and waits for that moment to complete the remaining stages. So, primary oocyte completes the prophase 1 of the meiosis 1 and will be waiting for the right signal to complete the metaphase in telophase and produce the secondary oocyte and the first polar body. Similarly, the secondary oocyte, once it is formed, will be completing the prophase 1 and the prophase 2, metaphase 2. That means the secondary oocyte, once it is entering into the maturation division 2, will complete the prophase 2 and also the metaphase 2. So, it will complete that two initial stages of the karyokinesis, and once the sperm enters into the secondary oocyte, which has already completed the metaphase 2, the remaining stages by name the anaphase, the telophase, the karyokinesis is done, and that is followed by the cytokinesis, and we get the structure called the ovum and the second polar body. So, it is a very unique feature in the process of oogenesis, where the primary oocyte will complete the prophase 1 and remaining part later. Once the secondary oocyte is formed, it will complete the metaphase 2, and once sperm make its way, a single sperm makes its way into the cytoplasm, it will complete the anaphase, telophase, that is the karyokinesis, and that is followed by the cytokinesis, and ultimately there will be production of the female gamete called the ovum with the release of the second polar body, which is bound to get disintegrated. We have studied the process of oogenesis and understood that the secondary oocyte will become the ovum after the entry of the male gamete during the fertilization. Now, we want to know how this follicular development or the growth takes place in the ovary. The ovary, the female gonad, is divisible into several regions. So, when you demarcate within the ovary, we find the first one being the germinal epithelium, that is followed by the tunica albuginea, and that is followed by the entire stroma. The stroma divisible into outer cortex and the inner medulla. We find the growth of the follicle in the region called as the ovarian stroma, that is the cortex. That means in the ovarian cortex, which is the outer component of the stroma, we find the various developmental stages of the follicle. So, let us study what are the various stages which are involved, ultimately resulting in the making of the Graafian follicle. So, I start my explanation with the fundamental first part called the primordial follicle. What is a primordial follicle? Follicle itself always refers to a multicellular unit, a group of cells, and here when I say the term the primordial follicle, the very first one you can see here, in the center of the primordial follicle there is a cell, that is the primary oocyte, and the primary oocyte is surrounded by the squamous cells. So, basically the squamous cells in future will get modified, and we must understand that the primary oocyte will already complete the prophase 1 of the meiosis 1, and at that stage the further activity is suspended. Now, the primordial follicle is containing the primary oocyte and is surrounded by the squamous cells. Now, this will proliferate, this will grow, and we get the next stage called the primary follicle. I repeat, the primordial follicle will be converted into the primary follicle. What are the various cells which are present in the primary follicle? In the primary follicle, we have again the same primary oocyte, and the squamous cells which are here are becoming cuboidal. They are not only becoming cuboidal, but they also increase in their number. You can see this. These cuboidal cells are increasing in their number. This is about one layer, and probably repeated mitotic divisions. These cells increase in number. So, what was a squamous cell became a cuboidal cell, and the cuboidal cell is increasing in number, and the layerings will increase one above the other. You can see a multi-layer system, and these cells are called by the name the granulosa cells. Now, the granulosa cells are the diploid cells, and of course, the primary oocyte is also at the moment the diploid. The granulosa cells will increase in their number. Now, when you see the increase in number, see the increase in number is observed here, and also there is a formation of a covering which is called as the theca. Actually, the theca is the outermost layer of the follicle, and now with the theca, the inner granulosa cells, and in the center the primary oocyte, this particular multicellular body will be called as the secondary follicle. That means the secondary follicle is in its initial stages. So, the secondary follicle in its initial stages will have an outermost covering called the theca and inner granulosa cells, and of course, gradually there will be formation of a fluid filled space which is called as the antrum, and this is the primary oocyte. Now, this particular secondary follicle having the theca, the granulosa cells, and the primary oocyte will be transforming into a further upgraded structure which is having the parts like this. If you look at this parts of this particular multicellular unit, you can see the theca differentiated into the theca externa and the beneath which we find the theca interna. So, the theca externa and beneath which is the theca interna, externa, and then the interna, and that is followed by again the granulosa cells. Now, these granulosa cells, the terminology can be membrane or the membrana granulosa. This is what is called the membrana granulosa. We can say so too many cells, and which we previously called it as the granulosa cells, like the theca externa, theca interna, and the granulosa cells. You can see one important region here called the antrum. The antrum is a fluid filled region whose dimension will be increasing. So, the fluid filling will be increasing, the dimension of the antrum will also increase. As you go to the inner cellular arrangement, when you see here, we can see a group of cells which are very close to the mother cell or inner cell. This is called the corona radiata. The corona radiata cells are actually resting on a glycoproteinaceous layer called the zona pellucida. That means the zona pellucida, which I am indicating again, look at this small layer, zona pellucida, surrounding the zona pellucida, we find the diploid cells very closely associated called the corona radiata. So, corona radiata, then this is the zona pellucida, and as you see deep inside, the space is called the perivitelline space, but I have written two cells there. That is, one of them is called the secondary oocyte, other is called the first polar body. This is an important suggestion here, that when the primary oocyte is becoming the secondary oocyte and releasing the first polar body, It means that the primary oocyte has completed the meiosis 1. This transformation of the primary oocyte into the secondary oocyte with the release of the first polar body is a very important crucial moment, because the diploid cell became the haploid. I repeat, the diploid cell, the primary oocyte, became the haploid cell, and another haploid cell, which is nothing but the first polar body, which is definitely a product of unequal cytokinesis. Now, when this is formed, when these haploid cells are formed, we see that the surrounding cells here attain the status of endocrine cells. So, endocrine production called the estrogen will be beginning. So, the theca interna and the other the cells here, the follicle cells here, will become endocrine, and they will release the female sex hormone, which is called as the estrogen. So, this transformation of the secondary follicle into the next one can be termed as the tertiary follicle, and the tertiary follicle will further increase in the number of cells, the size, and that is what is called as the Graafian follicle. So, let us see this important transformation one more time. The primary oocyte is becoming the secondary oocyte and the first polar body because of the meiosis 1. The theca demarcated into the theca externa and the theca interna, and the granulosa cells will be clearly demarcated into the membrana granulosa cells and also the corona radiata cells. But the corona radiata cells are very closely present on the glycoproteinaceous layer called the zona pellucida. So, from outside to inside, maybe I will take from the direction of the antrum. So, the sequence will be theca externa, theca interna, and the granulosa cells, and again the antrum, and again you will get group of granulosa cells, and here is the corona radiata, zona pellucida, perivitelline space, and ultimately the secondary oocyte. I repeat this: theca externa, theca interna, membrane granulosa, and antrum. If you take this direction, antrum, again the corona radiata, zona pellucida, perivitelline space, and the secondary oocyte will be the sequence for the tertiary follicle, which will further grow to become the Graafian follicle. The various stages in the follicular development, as observed before, will be like the primordial follicle, primary follicle, secondary follicle, tertiary follicle. When it is fully grown, we can term it as the Graafian follicle. I want to completely present before you the picture of the Graafian follicle, which is ready for rupture. The theca externa, the outermost covering, and the second one, theca interna, which is endocrine, and will be releasing the estrogen, the steroidal hormone, the female sex hormone, and that is followed by a layer of the membrane granulosa comprising of the granulosa cells, and then we have this fluid filled space, which is called the antrum, and then this group of cells called the corona radiata resting on zona pellucida, the perivitelline space, and here is the secondary oocyte and the first polar body. Now, at times we use a word called cumulus oophorus. When you take this word called oophorus, oo stands for the egg and the phora standing for the support. Now, because the antrum size will be increasing, there will be a kind of dislocation or the change in the positioning of this unit. The secondary oocyte with the zona pellucida and corona radiata will be slightly moving away, and we call it as eccentrically placed secondary oocyte. At that time, when it is moving away or present very away from the center, eccentrically placed structure, then this group of cells which are here, which are supporting, sometimes are called by the word cumulus oophorus. So, this is Graafian follicle, and what is important here is all are diploid cells here, all are diploid cells except for the secondary oocyte and the first polar body. That is one important point to be noted. Second is the release of the hormone, that is estrogen, done by the endocrine cells of Graafian follicle. So, when the levels of estrogen gradually increase, it will act like a signal for the release of the luteinizing hormone. So, estrogen levels, when they increase, that will be signaling the release of LH, which is the luteinizing hormone, a gonadotropin and a product of adenohypophysis. The luteinizing hormone will act on this fully grown Graafian follicle, and that will result in the rupture of Graafian follicle. So, the sequence of endocrine signaling will be the release of estrogen, the increase in the level of estrogen acting like a trigger for the release of LH, and LH is the luteinizing hormone, a gonadotropin, a product of adenohypophysis. That LH will be released in excess quantities or large, and we call it as LH surge. And this LH will act on the Graafian follicle, and LH, once it acts on the Graafian follicle, it results in the breakdown or rupture of the Graafian follicle, and as a result, the central body, the secondary oocyte, the first polar body, the zona pellucida and the entire corona radiata, this is released from the Graafian follicle, and the release of the secondary oocyte from the ovary due to rupture of Graafian follicle is called as ovulation. That means LH is causing ovulation by acting on the Graafian follicle. Once ovulation takes place, the secondary oocyte, the polar body, the zona pellucida and corona radiata, that will be released into the body cavity collected by the fimbriae of the fallopian tube. And now, once the rupture is done, the group of follicle cells which remain in the ovary will be called as the corpus luteum, and the corpus luteum begins to release a hormone called progesterone. So, the very name called luteinizing hormone will suggest that the luteinizing hormone will be certainly meant for making of the corpus luteum. So, LH acting on Graafian follicle, the Graafian follicle rupturing, the release of secondary oocyte called ovulation, and the formation of corpus luteum. So, LH will cause both formation of corpus luteum and also the ovulation, which means nothing but the release of the secondary oocyte. Now, let us learn the structure of sperm and ovum. We have studied the gametogenesis, namely spermatogenesis and the oogenesis. Spermatogenesis ultimately results in the male gamete called the sperm, and the oogenesis, if it is completed, there will be formation of ovum. The important point to be noted is the secondary oocyte will already complete the metaphase 2, and after the penetration of the sperm during fertilization, the secondary oocyte will complete the remaining 2 stages of karyokinesis, and that is followed by the cytokinesis, producing the ovum and releasing away the second polar body, which will disintegrate. Now, that means that completion of spermatogenesis will finally produce the sperm, whereas oogenesis will be completed after the entry of the sperm into the secondary oocyte. Now, we are going to study the structure of the sperm. The male gamete or the sperm will be having the structures like this. It has a head, and then that is followed by the neck, the middle piece and the tail. Now, this particular part called the tip of the sperm or the structure which is anterior to the head, called as the acrosome. This will contain a derivative of the Golgi, which is a vesicle rich structure. It is containing several enzymes, and one of the enzyme is the hyaluronidase that is present within this vesicle which is part of the acrosome. So, the tip of the sperm is called acrosome, which is containing a vesicle which is a derivative of Golgi, and that is enzyme rich, and one of the enzymes is called as the hyaluronidase. Now, the head is containing the nucleus. This is the haploid nucleus, and it will have 23 chromosomes, but it can be 22 plus X or it could be 22 plus Y. That's why when we understand spermatogenesis, 50% of the sperms will be carrying the X chromosome and 50% of the sperms will be carrying the Y chromosome. But the total number of chromosomes in the nucleus of a sperm will be 23, and that is followed by the neck. The neck is followed by the middle piece. The middle piece is containing the powerhouse of the cell by name the mitochondria. So mitochondria will be present within the middle piece, and the formation of the mitochondria sometimes can be called as the bunkan, and this is followed by the tail. So tail is the end part of the sperm, which is meant for the movement, and the dynamics of the sperm is because of the tail, and the energy for the tail movement is given by the mitochondria which are present in the middle piece. And this tail is again a kind of eukaryotic structure and will have the 9+2 organization and is a derivative of a basal granule. So the various components one more time will be head, neck, middle piece and tail, and the nucleus is 23. Either it could be 22 plus X or it could be 22 plus Y. Now coming to the ovum, as I was referring to, after the entry of the sperm, the secondary oocyte will be transformed into the ovum, and within the cytoplasm of the secondary oocyte, the activity will be suspended near the metaphase 2. Once the sperm enters, the NR-tilo will be completed, cytokinesis will be completed, and ovum will be formed, and that is followed by a syngamy or fertilization. Here I present the basic structure of the ovum. The ovum is having a nucleus and which is always 22 plus X, and this portion is generally called the animal hemisphere. This portion is called the vegetal hemisphere, and this structure after fertilization will be called as the zygote and will continue the embryonic development. We are observing the development of the follicle at its various stages of development. This one is the primordial follicle, consisting of the primary oocyte surrounded by the squamous cells. The primordial follicle will grow in size, a cellular number also will increase, and that is what is called the primary follicle. As you can see here, it is called growing follicle, means the primary follicle, the secondary follicle, and here we can see the maturing follicle, and finally in this picture we can see the Graafian follicle with a very well developed and enlarged antrum. This darkened portion is the antrum. You can see the gamete which is eccentrically placed here, and once it is ruptured because of the LH, there is formation of the corpus luteum, and in the absence of pregnancy, the corpus luteum will be converted into a degenerate structure which is called the corpus albicans. This picture is to be notable, a Graafian follicle, and of course there is no egg here, it is actually the secondary oocyte, and after rupture, formation of corpus luteum releasing progesterone, and ultimately in the absence of pregnancy, the luteum degenerates to form the corpus albicans. After understanding the spermatogenesis and the oogenesis, let us throw some light on the various questions. Here the question number 1, which one of the following is not the component of the semen? The emphasis is on the word not the component of the semen. If you look at the options here, prostaglandins, citric acid, antrum and fructose. See, prostaglandins are certainly the component of the semen, and these are released from the seminal vesicles of the male reproductive system, that is the accessory glands of the male reproductive system. So naturally semen will contain the prostaglandins. Coming to citric acid. Citric acid is also one of the component of semen released from the seminal vesicle, and that is utilized by the sperm for the oxidation. And then what about the antrum? Antrum is not the component of the semen or the seminal plasma, because antrum is a fluid filled space that is found in the tertiary follicles and also the Graafian follicles, and fructose will be one of the component of the semen, which is used for cellular oxidation and the energy production. So, the answer for the above question, which one of the following is not the component of the semen, the answer would be antrum. The second question is about select from the given list the diploid cells. It is important for us to understand that the moment the cell begins the meiosis 1, the diploid cells will be changing into the haploid cells. For instance, the primary spermatocyte, if it does the meiosis 1, it will become the haploid secondary spermatocytes. So, meiosis 1 is a very important moment wherein the cells will become haploid from diploid. So, the question here is, select the diploid cells from the following. Spermatogonium. We all know that spermatogonium is a mother cell, sperm mother cell, and so it will be diploid. So, A is a diploid cell. So, that is possible. Then, first polar body. The first polar body is formed from the primary oocyte after meiosis 1. So, it will be a haploid cell. So, this is not correct. Then what about secondary oocyte? The primary oocyte after meiosis 1 will produce secondary oocyte, haploid, first polar body, haploid. So, secondary oocyte is haploid. Then again, spermatid. We know that after the primary spermatocyte, meiosis 1, secondary spermatocytes, the meiosis 2, there will be formation of spermatids and they are haploid. So, these are also not the diploid. So, the last one, the oogonium, similar to the spermatogonium, oogonium is also a diploid cell. So, this is also diploid. See, out of the given list, here spermatogonium is diploid, first polar body is haploid, secondary oocyte is haploid, spermatid is haploid, and oogonium is diploid. So, the answer is the spermatogonium and the oogonium. So, it is A and E are the two cells which are diploid. Which one of the following is not a cellular layer? See, in the options given here 1, 2 and 3 and 4, the various components of the Graafian follicle are mentioned, and the question is which one is not a cellular layer. In fact, entire Graafian follicle will have complete cellularity, multicellular structure and with diversified cells. Now, the only one which is not a cellular layer but a biomolecular layer is the zona pellucida, which is a glycoproteinaceous layer. So, the answer would be the zona pellucida, which is a glycoproteinaceous layer, and this is the one which is not a cellular layer, which forms a layer beneath the corona radiata. That is what is called zona pellucida, which is not a cellular layer, it is a molecular layer, glycoproteinaceous layer. Remaining all, the theca interna, granulosa cells and corona radiata, all these three are the cells, and in fact, they are the diploid cells of the Graafian follicle. So, the answer is the zona pellucida, which is not a cellular layer.